Welcome to this review of Veterinary Parasite Taxonomy. Hi, I'm Christopher. In parasitology, names matter. They show up in textbooks, diagnostic reports, product labels, treatment plans, and clinical conversations. But those names become much easier to learn when we first understand where they fit. So in this video, we're building the map. Parasites are incredibly diverse. They infect different hosts, live in different tissues, move through different environments, and belong to very different branches of life. Once we understand the major groups, the details become easier to organize and apply. The goal today is simple. See the big picture, so the names, life cycles, and diseases have a place to land. Parasites come in extraordinary sizes and forms. Some are microscopic, like protozoa that require a microscope to see, and others are massive, like tapeworms that can reach extraordinary lengths in marine mammals. That range tells us something important. Parasite does not describe one body type or one evolutionary family. It describes a way of life. So before we learn individual parasites, we need to organize the major groups. Here is the full taxonomy map. At first glance, it can look intimidating. That's normal. Taxonomy diagrams have a way of looking like someone spilled Latin into a flowchart. But we're going to build this step by step. The most important idea is that veterinary parasites fall in several major practical groups: arthropods, nematodes, platyhelminths, acanthocephalans, and protists. Some of these are true evolutionary groups. Others are more practical teaching groupings. This map is useful, but it is not meant to be absorbed all at once. The first major split is between animal parasites and protist parasites. Both sit within the larger eukaryotic world, but they occupy very different parts of that world. Animal parasites include groups like arthropods, nematodes, flatworms, and thorny-headed worms. Protist parasites are microscopic eukaryotes that are not animals, plants, or fungi. This split is one reason parasitology feels broad. We are not following one tiny family tree. We are building a practical veterinary map. What is a parasite? For this course, we will use a practical veterinary definition. A parasite is a eukaryotic organism, usually a protozoan, helminth, or arthropod, that lives on or in an animal host and benefits at that host's expense. That definition is intentionally more narrow than some broad biologic definitions. In a broader sense, viruses, bacteria, and fungi can behave parasitically. But in veterinary parasitology, we usually separate those into other disciplines. Our focus here is protozoa, helminths, and arthropods. So what is a eukaryote? A eukaryote is an organism whose cells have membrane-bound nucleus and other membrane-bound organelles. Animals are eukaryotes, plants are eukaryotes, fungi are eukaryotes, protists are eukaryotes, and you are a eukaryote. Bacteria and archaea lack membrane-bound nucleus and are called prokaryotes. This matters because animal hosts belong within eukaryota, and so does veterinary parasites we're focusing on in this video. A dog, cow, horse, cat, bird, fish, and human are all eukaryotes. So are protozoa, helminths, and arthropods. Within the larger eukaryotic world, animals are one branch. Now, that may sound obvious, but it matters for taxonomy. When we talk about animal parasites, such as arthropods, nematodes, flatworms, and thorny-headed worms, we are still within the animal side of eukaryota. But protozoal parasites sit elsewhere in the eukaryotic world. They're all eukaryotic, but they are not animals. This is one reason veterinary parasite taxonomy feels messy. We are not following one family tree. We are building that practical map across several branches of life. We will start on the animal side of ecdysozoa, the molting animals. The name comes from ecdysis, meaning shedding or casting off, and zoa meaning animals. Ecdysozoa are literally animals that grow by shedding. When we discuss arthropods, we will use the term ecdysis for the process of casting off an outer cuticle or exoskeleton. If you have a friend named Zoe, the name comes from the Greek word for life. Same neighborhood of Greek word roots, slightly different usage here. This branch includes arthropods and nematodes, two of the most important groups in veterinary parasitology. We will begin with arthropods because they include ectoparasites, vectors, and several parasite-adjacent organisms that veterinarians need to recognize. The phylum Arthropoda includes jointed, exoskeleton-bearing ecdysozoans. In veterinary parasitology, the major arthropod groups we need to recognize are pentastomes, arachnids, and insects. Pentastomes, or tongueworms, look worm-like, but they are arthropod-affiliated parasites. Arachnids include ticks and mites. Insects include flies, fleas, and lice. Arthropods matter because they can be parasites themselves, but they can also act as vectors of disease. Now let's look at the insects. The core insect orders for veterinary parasitology are Diptera, Siphonoptera, and Pithecoptera. When trying to remember these terms, the etymology can be surprisingly helpful. Diptera means two wings. True flies have one functional pair of wings, while the second pair is modified into balancing structures called halteres. Diptera includes flies and mosquitoes. They may be biting pests, vectors, or agents of myiasis. Siphonoptera comes from the words meaning tube or pipe and wingless. That fits fleas nicely. They are wingless insects with piercing, sucking mouth parts. Fleas are ectoparasites and can serve as vectors or intermediate hosts. Pithecoptera means wingless lice. These are the lice, and the name reminds us that they are wingless ectoparasites. Lice are permanent ectoparasites of mammals and birds, and they are often highly host-specific. Then we have the parasite-adjacent insects. That includes Trichoptera, Blattodea, and Coleoptera. These may not be click parasites, but veterinarians still need to recognize them because they can be vectors, contaminants, intermediate hosts, toxin concerns, or lookalikes. Before we go deeper into worms, we need to pause on the word helminth. Helminth is a practical clinical term for parasitic worms, but it is not one tidy evolutionary family tree. Nematodes are roundworms, and they belong within Ecdysozoa. Platyhelminths are flatworms, including flukes and tapeworms, and they sit outside Ecdysozoa. Acanthocephalans are thorny-headed worms. They are helminth-like parasites with their own distinct biology. So when you hear helminth, think practical clinical category, not one close evolutionary lineage. Similar shape, different biology, different clinical logic. Now let's add the second major ecdysozoan parasite group, Nematoda, the roundworms. Nematodes are important because they include many of the gastrointestinal, respiratory tissue, and vector-borne worms that veterinarians encounter across species. This part of the map helps us understand why roundworms sit near arthropods in Ecdysozoa, even though we often discuss them clinically as helminths. Nematodes are roundworms. They are unsegmented cylindrical worms with a tough cuticle, a complete digestive tract, and grow by molting. They include many of the most important internal parasites in veterinary medicine. In this overview, we introduce Nematoda using two major practical buckets, Enopleta and Caesarnentia. Enopleta includes trichuroid-type nematodes such as whipworms and capillarid-type parasites. Caesarnentia is a practical teaching group that contains many of the major roundworm orders that we will encounter. Within Caesarnentia, we organize many major veterinary nematodes into several orders. Strongylida includes strongyles and hookworms, with many gastrointestinal and some lung parasites. Oxyuretta includes pinworms, very important in horses. Ascaridida includes large roundworms, such as ascarids, many of which have important larval migration patterns. Rhabditida includes strongyloides-type parasites, including organisms with skin penetration and complex life cycle features. Spiroidea includes many tissue-associated and vector-borne nematodes, including filarioids and spiroids. The takeaway is that Cercinetia is a practical way to organize many of the nematodes that matter clinically. Next, we move to worm-like parasites outside of ecdysozoa, acanthocephalans, and platyhelminths. This is where the word helminth can be useful, but also potentially misleading. These organisms may be worm-like and clinically grouped within helminths, but they are not from the same evolutionary branch as nematodes. Acanthocephalans are thorny-headed worms. They are obligate parasites with retractable proboscis armed with hooks or spines. That proboscis helps anchor them to the intestinal wall of the host. They do not have a digestive tract, so they absorb nutrients across their body surface. They are not emphasized as often as nematodes or flatworms, but they are important in wildlife, fish, bird, and some domestic animal contexts. They also serve as a great reminder that worm-like does not mean the same biology. Platyhelminths are flatworms. The three major classes we introduce here are Trematoda, Cestoda, and Monogenia. Trematodes are flukes. They usually have indirect life cycles, and a snail commonly serves as the first intermediate host. Cestodes are tapeworms. Many veterinary tapeworms use one intermediate host, although some have more or less complex patterns. Monogenians are usually direct life cycle parasites, particularly important in fish. All are flatworms, but their life cycles differ dramatically. Now we will switch from animal parasites to protists. Protists are eukaryotic, but they are not animals, plants, or fungi. The protist side of the map is very diverse, so we will treat it as a practical framework rather than a simple kingdom. Protist is a practical term for a very diverse collection of microscopic eukaryotes. Some are free-living, some are symbionts, many are parasites. In veterinary medicine, protists matter because they include major intestinal parasites, tissue parasites, blood parasites, and vector-borne organisms. This part of the map is more taxonomically complicated, but the same principle applies. Use the map to organize clinical thinking. One major protist supergroup is SAR, which includes Stromenipiles, Alveolata, and Rhizaria. Before we go further, a clade is an evolutionary branch, a group of organisms that share a common ancestor and includes that ancestor's descendants. Unlike terms such as class, order, or phylum, a clade is not tied to one fixed taxonomic rank. Clades can be large or small, and they can be nested inside one another. So here, SAR is the larger clade, and Stromenipiles, Alveolata, and Rhizaria are major branches within it. Stromenipiles include a smaller number of veterinary relevant genera. Alveolata is a big veterinary branch here. It includes Apicomplexa, ciliates, and dinoflagellates. Rhizaria is important in a broader SAR framework, though less central to the main veterinary parasite groups we emphasize. For this overview, Alveolata is where much of the veterinary parasite action lives. Within Alveolata, Apicomplexa is a major veterinary group. Two classes are useful for organizing this branch: Conoidocida and Akynoidocida. Conoidocida includes many coccidian and tissue-forming genera, such as Eimeria, Cystoisospora, Toxoplasma, Neospora, Sarcocystis, and Hepatozoon. Acanthocephalans includes many hemoparasites and vector-borne apicomplexans. Pyroplasmida includes genera such as Babesia, Tyleria, and Cytoxoon. Hemosporida includes genera such as Plasmodium, Hemoproteus, and Leucocytozoon. These organisms are small, but clinically, they are anything but minor. At this point, the big map should be starting to look less like a wall of names and more like a working framework. We have built the animal side and started the protist side. Next, we will add 2 more protist groupings that include important veterinary and zoonotic organisms: excavata-associated protists and amoebozoa. Another practical protist grouping is excavata and related lineages. Here we introduce several veterinary and zoonotic groups. Kinetoplastia includes kinetoplasts such as Trypanosoma and Leishmania. Diplomonadida includes Giardia and Spironucleus. Trichomonadida includes Trichomonas, Tritrichomonas, Pentatrichomonas, and Histomonas. And Heterolobosea includes Naegleria fowleri, often called the brain-eating amoeba in public-facing language, although it is better described taxonomically as an amoeba-like Heterolobosean protist. This slide is a good reminder that common names in taxonomy do not always line up neatly. Amoebozoa includes true amoeboid protists with Lobopseudopodia. Important genera include Entamoeba and several free-living opportunists like Acanthomoeba, Balamuthria, and Sapenia. Some are associated with intestinal disease. Others are important because they can cause ocular or central nervous system disease under the right circumstances. And again, do not confuse amoebozoa with Naegleria. Naegleria fowleri is amoeba-like, but taxonomically it belongs within Heterolobosea. That one distinction is worth remembering. Now we come back to the big map. Hopefully it looks less intimidating than it did at the beginning. Veterinary parasites are eukaryotes, but they are not one tidy family. Arthropods include ectoparasites, vectors, and parasite-adjacent groups. Nematodes are roundworms within ectozoa. Platyhelminths and acanthocephalans are helminth-like groups outside of ectozoa. Protists are microscopic eukaryotes with several major lineages of veterinary importance. The point of taxonomy is not memorization for its own sake. Taxonomy is the map. Clinical logic is the destination. As you move through parasitology, keep asking the same questions. What kind of organism is this? Where does it live? How does it move? What host does it need? What stage can we detect? How does it cause disease? And how does it reach the next host? Those questions turn taxonomy into veterinary reasoning. I'm Christopher Lee. Thank you for watching. And what you know today helps protect everyone tomorrow.